Hello everyone, welcome to BrainMap. This seminar series is co-sponsored by the P41 funded Center for Mesoscale Mapping, housed in the Martinos Center. It's our great pleasure to have Dr. Nikolaus Weiskopf today to give us special BrainMap. Dr. Nikolaus Weiskopf is the director of the neurophysics department at the Max Planck Institute for Human Cognitive and Brain Science in Leipzig, Germany. Dr. Weiskopf is an expert in MRI method and their application to neuroimaging. His current research focus is to develop functional and anatomical microstructural image using MRI in order to study structure, function, and their interplay at the microstructural level. He received his PhD from Graduate School of Behavioral Science and International Max Planck Research School in Tübingen. In 2004, he did his postdoctoral training as a senior research fellow at the Wellcome Trust Center for Neuroimaging, University College London. In 2006, he became head of physics group at the Wellcome Trust Center for Neuroimaging at UCL. In 2014, he was appointed professor of MR physics and neuroimaging at the Institute of Neurology, UCL. Since April 2016, he has been a full-time director of the Department of Neurophysics. He holds an honorary professorship at the Faculty of Physics and Earth Science at the University of Leipzig, and an honorary professorship at the Institute of Neurology, UCL, United Kingdom. I would like to remind our audience to address any questions using Q&A box or raise their hands at the end of the talk. Dr. Weiskopf, thank you very much for coming today, and the stage is yours.
Thank you very much for the kind introduction. Can everybody hear ourselves properly? Yeah, looks like it's fantastic. Yeah, thanks a lot also for the invitation to give a BrainMap seminar here today. And I think it was already, I had a fantastic experience already. Spent a few hours here at Martinos Center, and I've seen all the fantastic facilities you have, the research that is going on. It's very impressive. And I can see also quite a few parallels in terms of the interests, looking at microstructure, looking at mesoscopic brain structures we're interested in, and also structure-function relationships. And my presentation will be exactly about that. We're going to look at what we pursue in the Department of Neurophysics in Leipzig, which is a lot about understanding the brain microorganization better in terms of its function and in terms of its anatomy or structural components. And so what I'd like to do is, we are interested in the functional and anatomical microorganization of the brain. And I'd like to say a few words about it. Initially, why we are interested and what it means, what we are looking at there, what drives our research interest. And in our institute in Leipzig, we're very much interested in what makes humans special. We're interested in cognitive neuroscience. We use cognitive neuroimaging tools quite widely. And the reason and one of the important aspects there is this question about that we know that structure determines function to a large degree, but we also know that training and functional changes can affect structure in turn. We have plasticity effects. So we have this kind of closed loop between structure and function and behavior that is mediated by plasticity and also developmental effects. And if you want to study that in humans, you need a non-invasive technique. And we believe that MRI is really currently the best technique out there that enables us to look at all these different aspects. But the challenge is obviously, at the moment, we are very limited in terms of the access to microstructure with MRI. As you know, I mean, it has been a great success story, looking at larger brain areas, the macroscopic view of the brain. but it's really useful to make yourself or remind yourself of how many spatial scales you have actually in the brain. So we are typically looking at this macro scale with MRI and function MRI, but there's a whole host of features at the micro scale, down to a micrometer or below. If you think about, for example, axons, thinking about cells, synapses, even smaller than that, and in between, stacked in between, we have the mesoscopic scale that is really important for function, mediates important functions when you think about, for example, cortical layers. You think about columnar structures that reach through the cortex. They are all functionally and anatomically relevant. And it would be fantastic to have full access with MRI to them. I think we're doing quite well with high-field MRI, 7 Tesla, to get to the mesoscale, but the microscale, we don't have direct access to it. We'll probably never have direct access with MRI in vivo. Thinking about, micrometer voxel size or so in vivo, very unlikely. But what we can do is we can build the right and appropriate biophysical models that allow us to get from the rich MRI data we have to glean information about the microstructure, to build models that give you something like a summary value on aggregate measure of something like axonal diameter, when you think about diffusion imaging, about myelin concentration, and so on. But you have to build the right biophysical models for that. And there are different approaches towards that. The modeling strategy we pursue is largely based on the fact that MRI makes it really easy to acquire images with very different contrasts that are sensitized to different aspects of the underlying microstructure because of different interaction effects, there are physical interactions happening there. And usually it would be difficult to get from one of those images then an estimate of the underlying microstructure, like, for example, myelination or iron concentration or other aspects. But by having different viewpoints onto the underlying microstructure, you can combine them and help with this inversion problem, going from MRI to the actual, let's say, biological metrics you are interested in. So our model is very much, or modeling approach is based on a multi-contrast approach. In addition, on quantification of MR contrast. So we use quantitative MRI a lot, and then with the right biophysical models, we try to make inferences upon the underlying biological microstructure. And in the second step, we are very much interested in to use this microstructure information, feed it into other biophysical models of function. This could be formal neuronal mass models, or it could be correlation analysis with fMRI, for example, to close the loop. I have been presenting at the beginning of the presentation about this plasticity link of structure and function and behavior. So this is kind of the general approach. In terms of the technologies, so the three-pronged approach we choose there. We work on the MR physics, increasing the resolution, signal to noise, lowering artifacts. We use biophysical modeling to get specific tissue information about parts of the tissue that are much smaller than the voxel size. It also helps us to standardize data across time points and sites. And then last but not least, we have a third pillar, which is proof of concept studies, proof of concept applications, and validation approaches using in vivo methods, but also a lot of post-mortem comparisons. So this is kind of the general concept I was trying to lay out to you at this point. Now I'd like to go into different examples, and you can visualize it in a way that we are now going from the outside in the brain, from the cortex, deeper and deeper into kind of eventually we'll end up in the midbrain area, looking at the substantia nigra. And first, I'd like to talk about the cortex a little bit and some of the applications there. So what we do there, we base a lot of our experiments and studies on the so-called multi-parameter mapping method, which allows us to acquire so-called flash data or gradient echo, spoiled gradient echo data with different contrasts So remember kind of our modeling approach. You want to have different contrast weightings, T1, proton density, and T weighting. We also get phase images out. And with the right physical models, which we implemented in the openly available hMRI toolbox, you actually get these quantitative maps out here, or semi-quantitative maps. And I present that here briefly because this will come back at several points now in the presentation where we exploit these maps to understand more about the structure and its link to function in the brain. And when you go to 7 Tesla, can get fantastic images with about up to 400 micrometer resolution of the brain. And I hope you can appreciate the very different contrast these different parameter maps offer, and the high detail, explicit contrast you get in the cortex, here cortical brain matter, for example, depicted by the R1 maps, longitudinal relaxation rate maps. Or here you can see in the R2-star maps that are sensitive to iron. You see the basic ganglia, the midbrain structures really well that have a high iron load. And this is only possible, obviously, at such high resolution that by dealing with the artifacts. For example, head motion is suppressed by optical prospective motion correction. Use also phase navigation to reduce respiration-related artifacts in these high-resolution images. And even at lower field strength, here this is data these are results we applied at 3 Tesla with Martin Serino and Frederick at the time in London, still, where we were constructing quantitative R1 maps from R1 measured inside the cortex as a proxy for myelination of the cortex. And you can see this on the inflated brain. All the bright spots are basically where the R1 values are high and myelination is expected to be high. And what you can see here, I put this here as an example. I think what you can nicely see is obviously the primary sensory motor areas pop out, and you can demarcate, delineate brain regions based on the myelination content measured non-invasively with MRI. And you can combine this, for example, with functional MRI, here with retinotopy, retinotopic analysis, using fMRI in the same subjects. And for example, what is striking, you can see this nice correspondence between the border of V1, delineated by this high myelin to low myelin contrast that overlaps very nicely with the borders of the primary visual cortex as identified by retinotopy. And so this is a nice example for studying structure-function relationships using one of these quantitative MRI tools. And we think clearly a step towards the in-vivo histology. These were early days, beginning of this type of research. Over the years, this has been taken much further. And one example I'd like to show here is taking this idea further in the sense that you can actually zoom into brain areas now. Rather than looking at the entire V1, we were, for example, looking at the secondary visual cortex. Daniel Henne, PhD student in our department, did that. And he started off from the tripartite scheme that the secondary visual cortex can be basically split into 3 types of stripes, thin, pale, and thick stripes that support different functions. And we also know that they show different histological properties in terms of cytochrome oxidase staining, but also myelin staining, so different myelin concentrations in these stripes. They're fairly small. We talk about 2 millimeters, roughly here in this direction in extent. And we would expect a clear structure-function relationship there. We know this from animal studies, but in humans, there's still quite a few open points about that. And that's why Daniel set out to study that further using these non-invasive techniques now. So what did he use? He used functional MRI and exploited the fact that the stripes are differentially sensitive to contrast. So that's what the thin stripes basically process and pick up primarily. And the thick stripes, they pick up the depth contrast, the stereoscopic contrast. And by presenting the right stimuli, you can identify these different stripe types now. This is actually collaboration with colleagues here at MGH in Boston, China, NASA, and Patel. And when when you've done the fMRI, you can use that to define regions of interest of thin and thick stripes by thresholding these maps. At the same time, then you acquired quantitative MRI data, one after star data, you can see over here, which serve as a proxy marker for myelination. And what you can do now, based on the functionally defined regions of interest, you can ask the question, do I have different levels, for example, of R1 in these different stripe types, which would indicate different myelination levels? And here you can see one example for the fMRI, color contrast, which identifies the thin stripes in the inflated and flattened brain here in the visual cortex. And here you can see the stripes in the secondary visual cortex. This is the primary visual cortex V1. Thick stripes you can't see as well as the thin stripes because it's more complicated to selectively stimulate them. Now you can expect the ROIs from that, and you can look at do the R1 values actually differ? And indeed, that's exactly what we found. When you look at this graph, these are the differences in R1 between here the thin stripes and here the rest of V2 of the secondary visual cortex without the thick stripes. So this would be the pale stripes, what you would expect. What you can see is that the R1 values are indeed lower in the thin stripes than in the pale stripes. And the same is true for the thick stripes. The R1 values are again lower than in the pale stripes, which we interpreted as a lower myelination of the thin stripes and thick stripes compared to the pale stripes based on the tripartite model. And this was actually reproduced. So these R1 values were estimated from multi-parameter mapping, but we also acquired MP2H protocols and estimated R1 values from there, and you see the same pattern. And what I would like to point out is also we varied the Z-score threshold for defining the ROIs on the fMRI data. And you can see that it's largely independent of the Z-score threshold. So it's not biased by selecting a particular threshold for the ROI definition. And I think this is a nice example for that you can really dig deeper with these techniques now and understanding now the mesoscale of the brain using the in vivo non-invasive MRI techniques. Now, when I was talking about R1 values, so the longitudinal relaxation rate being a metric for myelin, I was kind of glossing over an important fact that myelin is actually not a monolithic structure. When you look at myelin, I mean, it contains a lot of water, proteins, and a lot of lipids. It's actually a mixture of different compounds. And I think this is important to keep in mind, you know, when we use these shortcut descriptors and labels. And we thought it would be interesting to look a bit further into that. How does this composition of myelin, for example, change? And it was motivated by some obvious observations when you look at, for example, staining in histology, or you look at post-mortem imaging of the same tissue here, you can see that all of these stains stain for myelination, but they look very different. And why is that? Because they are differently sensitive to different subcomponents here. So there's a lot of potential wealth and richness in terms of information hidden in this composition of myelination. And it would be great to understand that better and exploit that actually. Similarly, you can see that these MRI images look quite different. So they might carry some of this information. So a better understanding of the complexity could help us greatly for potentially extracting interesting information. And one way to look at this is study Ilona Lip and Evgenia Kirillina were mainly pursuing is to, in particular, look at the lipid composition of myelin across the cortex, across white matter, gray matter. And for that, they used MALDI-Mass Spectrometry Imaging, which allows you to determine the different masses of the lipids in every pixel in your sample, and get for every pixel in your sample kind of this spectrometric analysis. And these different peaks, they represent different classes of lipids. And what you can see down here, if we pick out some of these peaks, you can see the maps here of the specimen, they look very different. So it clearly shows that different lipids in the myelin, and the lipids in myelin are differently distributed. Even within cortex, across layers, you have different relative contributions of lipids. And what we've also learned is by looking at different specimens here from top to bottom, that now the lipid peak height is indicated in colors here and brightness. You can see that across specimen, it actually looked relatively similar. It was relatively consistent, the composition. And there was a clear difference here between gray matter areas and white matter areas. For example, white matter areas tend to have lipids with higher masses. And that consistency led us to the point that we said it would be really interesting to see if we can learn something from that and if we can employ that in a modeling approach. And let me walk you through that slide. So what we've done here is at the top, you see quantitative MRI maps acquired post-mortem on a tissue sample. And then we trained, or basically we used a general linear model and also a simplified version of that to just predict the MRI contrast here from the spectrum of lipids, from these kind of spectra in every voxel. And when you do so with this linear model, down here, you see the modeling results. When you put all the lipid information in you have, you can actually see that the prediction solely based on MALDI, there's no MRI involved here, actually looks pretty much like the MRI data you've actually acquired. So it does a very good job. If you simplify now and you pretend there's only one type of myelin, so you reduce that to the first principal component, you can see you're doing a much worse job in terms of predicting the MRI data. And this is interesting for several reasons. First of all, it's great to see that even with just one component, just one myelin concentration metric, you're doing a relatively good job. That's promising for us. But including all the lipids gives you in additional information, in particular here in the R1 maps. They are much better predicted here by using all the lipid peaks, which tells you another thing. It means R1 is sensitive to the lipid composition, and maybe we can exploit that to do something like lipidomics in vivo. And here's the quantitative analysis. If you take all lipid peaks here, you explain much better the variability or the variance in the MRI data than using just the first principal component. So I think this bodes well, for an in vivo lipidomics approach there, but there's still a lot to learn. Now what I'd like to do is switch from the cortex into the superficial white matter. So go a little bit deeper in the brain. As I said, we're going from outside in now. And so why are we interested in the superficial white matter? Well, the superficial white matter is the structure that sits right underneath the cortex here, and it contains the short association fibers. You might have heard about the term U fibers, because a lot of these fibers connecting adjacent gyri, they follow the gyrification, so they look a bit like an U or inverted U. We are interested in those because if you just look simply at the numbers and you count the numbers of fibers in the white matter, then you have many more fibers in the superficial white matter than in the deep white matter. And we believe this tells you something about the importance of these fibers, in particular for the cortical-cortical connectivity, but also in terms of development processes, plasticity processes. The problem is when you try to run these experiments in humans, you're encountering this issue that you can't use the invasive techniques. You need to use MRI, non-invasive techniques, but we have very complex MR contrast mechanisms we don't fully understand. We know that when we do diffusion imaging, we have this problem that the short association fibers here run tangentially. The afferent and efferent fibers going in and out of the cortex run orthogonal to them. We have a crossing fiber situation, which is always kind of a complex situation for diffusion imaging. And we know this is a thin stripe of about 0.5 millimeter thickness, the superficial white matter. So we need very high resolution to image it. So a real challenge for MRI. But Evgenia Kirillina in the department, she was not discouraged by that. So she started looking at superficial white matter using very high resolution 7 Tesla susceptibility sensitive acquisitions like R2 star maps, so the parent transverse relaxation rate maps, or susceptibility maps. And when you look at them more closely, and others have done so as well, and before us, I should say this, you can see that there's a conspicuous bright high R2 star stripe or high susceptibility stripe just underneath the cortex. And that points towards that this is the superficial white matter. And because you have these higher susceptibility values, this points towards the paramagnetic substance driving the contrast. And we suspected iron to be the contrast driver. And we wanted to understand that better so we can better interpret what we see in the images. And so what Evgenia did, she went now from in vivo to post-mortem again here, looked at a temporal lobe sample here, imaged that post-mortem, the R2 star maps. You can see again this conspicuous bright stripe here, high R2 star values, but at the same time, she could take the samples and perform mass spectroscopy imaging on that, which tells you directly about iron concentration in every pixel, but also indirectly about myelin concentration. So this is for mass spectroscopy imaging. And what you can do now is actually, you can already see that by eye, I think, that obviously this stripe here is driven by a high iron concentration and not by a high myelin concentration. So over here, you can see the profile going from the pile surface towards a deep white matter here. And here going through the superficial white matter, you get a peak of iron concentration, which drives the peak in the R2 star, but you don't see a peak in the myelin concentration. So what does that tell us? I think it opens up fantastic opportunity in a sense that now we have potentially here a marker that is sensitive to the superficial white matter and different from the deep white matter and obviously different from the cortex. So it's exactly what we want to have, right? We want to understand superficial white matter better. And you can do this even in a quantitative way. You can take these two maps and predict the R2 star map from that quite accurately. But what you still don't understand is you don't see the microstructure in terms of cells and fibers here yet. And so Evgenia went and brought out the big guns, proton-induced X-ray emission, and also later on X-ray fluorescence. Well, they went to Hamburg to this 2.5 kilometer long electron synchrotron to do X-ray fluorescence to look at cellular and subcellular ion distribution now. And when you do so, you zoom into the superficial white matter, what you can see is that the iron is mainly in the somata of the oligodendrocytes and in the fibers themselves. And this is crucial because you can use this information now for modeling, for quantitative modeling. And it also tells you something about that this is the iron-sensitive aspects of MRI are directly related to basically the fibers in that domain. So we were now at the point that we could interpret the R2 star contrast in the superficial white matter as a marker for fiber density and fiber maturity, because iron concentration also changes with development. And you can go back now from post-mortem into the in vivo case and run a study there. Again, high resolution imaging, 500 micrometer resolution, and you can map the R2 star just underneath the cortex as a marker for the superficial white matter, as you can see up here. And you can look at the distributions now, and you can compare, for example, these distributions against the intracortical myelin distributions in the group. And what is quite interesting, you can see, for example, higher superficial white matter markers in the association areas, also in the frontal areas, where you would expect more U fibers. And it's almost complementary or almost anti-correlated to the intracortical myelination. We also know, for example, we have fewer U fibers in superficial white matter in the primary visual cortex, which also was picked up by that approach. So this now offers you a way to look non-invasively at superficial white matter using MRI in this particular marker. But what it does not do, so it gives you local information about the superficial white matter, but what it doesn't do is it doesn't give you information about the connectivity of superficial white matter. You might have realized that because you're looking just at point by point at the superficial white matter. And to look at the connectivity, we went from 7T down to 3T, but to very high gradient amplitudes. So we use the Connectom MRI system at our site. You know it really well. You get your next instantiation now at your site. Very exciting development here, the Connectom 2.0. This is Connectom 1.0 still, but I think gives you a fantastic opportunity in terms of pushing image quality, pushing image resolution. And to look at the superficial white matter and the short association fibers in there, what you need is resolution and image quality. And we further improved it for the occipital cortex by using these flexible surface coils, multi-channel coils. And here you can see some fiber tracts in the superficial white matter of the occipital cortex. So that worked really nicely well. And that allowed us now to go back and play the same game again we played before. We wanted to know how is function related in or mediated by the superficial white matter and the short association fibers. So what we do is we run high resolution tractography studies on the same individuals and perform on the same individuals functional MRI experiments. And since we love the visual cortex, it's well understood, well studied, we looked at the visual system initially as the first model system. And what you can do there is you can determine, for example, the retinotopy, this particular kind of topological organization of the visual cortex that aligns with the outer visual field of view. For example, you have eccentricity values going from the center of the visual field of view to the external periphery, and that is directly reflected in the cortical organization as kind of an easy transform between the two. You can also use the retinotopy to define the primary, secondary, and tertiary visual cortex in each individual. And what you can do now, that's interesting, you can say, well, look, I know the retinotopy, the eccentricity mapping, for example, here in the primary visual cortex, and I can ask the question, I now follow going from the primary visual cortex to the secondary visual cortex. How is that retinotopic information in these fibers translated from V1, primary visual cortex, to secondary visual cortex? And when we look at these short association fibers here in the same individuals, what you can see is this eccentricity information here shown in colors is actually preserved in the short association fibers. It's not like that it's completely garbled up, mixed up. It's actually preserved and directed, or you could say almost like parallel transported from V1 to V2. And this is a hierarchical system, right? The visual system. So you have a lot of information going from V1 to V2 and also backwards. So it's important to have these connections. So we were actually almost surprised to see this, to be honest, because we weren't quite sure about how reliable and how high would be the fidelity of the fiber tracking. We talk here again, 800 micrometer resolution. You need to follow these very thin fiber strips here in the superficial white matter, the short association fibers. And we were very excited about that. So we looked into it also in the postmortem sample to see how reliable are these tracts. And here's an example. You can see the in vivo tracts. Here you can see postmortem in a different subject, obviously, specimen. And you can see that the features are quite well preserved, even though the resolution here is much lower than the 200 micrometer resolution in the postmortem situation. And so you can then, this kind of motivated us to really take it one step further and ask the question, well, if it transports information from V1 to V2, how well does it? And you can do the following experiment. These are the actual eccentricity maps acquired with fMRI in V2 and V3 here. And down here, what you can see are the predicted retinotopies by taking the retinotopy in V1 and then following the short association fibers into V2 and projecting that into V2. So there's no information, no functional information in V2 here. This is solely based on functional information in primary visual cortex, plus the connectivity patterns through the short association fibers. And it's striking, the similarity you get. I mean, this is highly predictive here of these models. Just following the short association fibers are highly predictive of the actual acquired retinotopy. And you can even go from V1 to V3, jumping over one of the hierarchies, again, the features are really well reflected, and you can do this and plot it in a you can plot the eccentricity measures in V1 versus V2 in a scatter plot, and V1 versus V3, the predictions, so predictions versus the actual measurements, and you can see how well you do. I mean, we talk about predictions in the range of about a degree of visual field. It doesn't work as well, and I would like to point this out when you do this for polar angle, another feature of your retinotopic system, and this has to do with probably issues due to chiral biases in the tractography itself. Just wanted to make that clear. And the last thing I'd like to show on that is kind of a bit of kind of glass ball gazing, what we might be able to do with this kind of approach. I mean, it's fascinating. You don't need information in V2. So if you don't have it, for example, you could just predict it from V1, would be one example. You could probably already start thinking about pathologies where you could exploit it and look at it. Or you can do something like that, very popular in vision sciences, connective field mapping. So the understanding how different areas in the visual system connect to other areas. And we know all these different features of population receptive fields being different, connective fields being defined by functional MRI. That tell you exactly about how these areas connect locally. But what you can do with this approach now, you can supplement and extend connective field mapping, because with this approach, you can estimate how much of the sampling surface and increase of sampling surface, for example, when you go into V3 versus secondary visual cortex V2, is due to actually the short association fibers kind of fanning out. So you can determine one of the components that lead to the size of the connective fields. And this is due to the short association fibers. And when you look at it, this is what you would expect based on the functional MRI in terms of connective field sizes. You can see that in V2, they are lower or smaller than in the tertiary visual cortex, as you would expect, right? This is from functional MRI literature values. The contribution of the short association fibers is lower than that. And the difference gives you an estimate of maybe contributions of intracortical connectivity, dendritic tree connectivity that also lead to a certain spread. And the other thing we were quite excited about looking at that is that the measures you get out from diffusion imaging, that they are reasonable in terms of the order of magnitudes here. And they're also sensible in the sense that they are lower than what you would expect for the connective fields, because they're only one contributing factor that lead to kind of this spread of information across the visual field. So there's a lot we believe we can exploit for vision science and actually also other areas of the brain. And I'd like to show this very briefly because we have one department headed by Angela Frederici at the institute that is very much interested in language, in particular PROCAS area, has very specific ideas and hypotheses about intracortical connectivity in PROCAS area, but they didn't have the access to it so far. And we rolled out this super short association fiber mapping now into PROCAS area. And you can indeed now delineate connectivity between BA44 and BA45, the two constituting parts of PROCAS area. And what you can see, for example, when you look at this intra PROCA area connectivity, it differs between the left and right hemisphere. It's dominated by, or it has lateralization towards the left hemisphere, which makes sense in terms of language. And you can even extract fine-grained information about subdomains of the BA44 and 45 being connected. And we are currently working on pushing this even further out into larger studies. And here we also capitalize on a collaboration with MGH, bringing the G-slider method to look at the whole brain, combining that with optical prospective motion correction to now look at whole brain connectomes that go beyond the long-range fibers. So really looking at the short association fibers. Now, I'd like to just make a point. This might be obvious to most of you, but I think MRI is a fantastic versatile technique. So it doesn't mean you can only apply it to humans. It doesn't mean you can only apply it in vivo. It works post-mortem really well. You can apply it to other species. And here I just would like to show this project briefly, a research program, which is a collaboration that is across different Max Planck institutes, including the Evolutionary Anthropology Institute, and also includes university and also the Robert Koch Institute, which is kind of the German CDC, are involved in that as well. And what makes this project really special is we are looking here in particular at chimpanzee, but also other apes that have been monitored very closely by our colleagues at the Evolutionary Anthropology MPI over years, over decades, actually. They support these sanctuaries. And when a chimp dies there, then the brain would be extracted actually in the wild, in the jungle, directly fixed, sent to Germany. We do the MRI scanning, we do the histology there. Of course, to understand better, what makes humans special again, for example, language functions. What are the evolutionary trajectories? And I think MRI is a fantastic method because it allows us quick access to the entire brain, information about microstructural features of the entire brain. Down here, you can see some examples of these maps we generate. And here, again, a study led by Ilona Lipp, where she was looking at intracortical myelination measured by magnetization transfer and intracortical ion concentration measured by R2 star as a proxy, and looking at how that changes across the lifespan of chimpanzees. And this is special about the project because typically when you probably know there's a moratorium. So apes and chimpanzees are not scanned anymore because there's no experimental research on them anymore. And if we have access to tissue, it's typically tissue from this post-mortem tissue from zoos where animals become quite old. So we have really not a balanced age range. With this project, unfortunately, it's the case when you're in the wild, unfortunately, very young chimpanzee die. So we can study the lifespan. We can even look at wild versus captive animals. And so we could do a cross-sectional aging study, so to say, or lifespan study, and look at, how does myelin change with time? So look into these hypotheses about early versus late myelinating tissue. Are there differences to humans? Also, we could look at R2 star as an iron marker. And what we saw there, for example, is that they accumulate iron much more slowly than humans. And this is highly interesting because maybe it explains some of the difference in terms of neurodegenerative diseases. And you can also look at differences, of course, of the myelination compared to humans indirectly. And you can see that the frontal brain areas tend to be relatively more highly myelinated. So there are a lot of potential evolutionary questions you can address with it, with this type of study and approach. And I think MRI is great for that type of experiments and research to answer these type of research questions. I didn't show the histology, but we also do histology, obviously, in the tissue. Now, the last example I'd like to show is now going even deeper into the brain, into midbrain structure, so-called substantia nigra. And this will be all about iron in the brain. So this is the substantia nigra, rather small, few millimeter sized structure, but not less important because of its smaller size. It actually plays an important role, we know, in certain neurodegenerative diseases, in particular in Parkinson's disease. We know when we look at the substantia nigra, we see these clusters of dopaminergic cells, so-called nigrosomes, in particular nigrosome 1 here, where you have a lot of dopaminergic cells. We know that about 60 to 70% of these cells have died before patient usually would go to the neurologist with motor symptoms and be diagnosed with Parkinson. So if we had a metric for measuring the cell density there, that would be non-invasively, we could apply early on, we would have potentially a therapeutic window for treating maybe Parkinson's disease early on and saving and salvaging some of the cells. What is interesting about these cells, they're fairly large dopaminergic cells, and they contain a lot of neuromelanin, which is a byproduct of dopamine generation and synthesis. And they are pigmented, so they look dark, like the melanin in our skin, basically. And they also contain a lot of iron, so they accumulate iron. And we thought, in particular here, Malte Bramelo, PhD student in the department, we thought it would be really interesting to understand the contrast mechanisms of MRI better in that area as a potential biomarker. And when you look at it, for example, again here post-mortem, and you scan with relatively high resolution, you can even see the dopaminergic cells here directly in the T2 star weighted image. They appear dark. This is consistent with a high iron concentration. When you do quantitative mapping, R2 star is increased here in the nitrosome. And when you zoom into it even further using this proton-induced X-ray emission, or PIXI again, you can look at single cells here and you can see the increase of iron concentration in the cells. You can do this across a larger brain area, and you can now correlate basically these fully quantitative PIXI maps with classic histology. So you can use now larger samples, just doing classic histology by cross-calibration. And you can use these larger samples now to really determine the iron distribution in the tissue at every point. And when you have the iron distribution at every point in the tissue, what you can do is you can make predictions about the B0 inhomogeneities, the Larmor frequency distributions, and they directly help you to determine and predict R2 star at every point, but also in a large tissue piece or larger voxel where you can't see the single cells anymore. That's important. So you can build these predictive models. And that's what Malte has done. And you can see here that the experiment here, the red dots, is actually really well predicted by the static defacing regime model. And don't want to bore you with the details here, but one of the upshot is, or the takeaway here is that when you measure R2 star maps and R2 maps, so basically 1 divided by T2 would be R2, and you subtract them from each other, so you get the R2 prime metric, you can predict from this model that about 60% of the R2 prime is driven by the iron in the dopaminergic neurons. And let that sink in, because I didn't say it's driven by iron. I said driven by iron in dopaminergic neurons. Because when you look at the substantia nigra, there's a lot of iron in these substantia nigra, not only in the dopaminergic neurons. But because it's so highly concentrated in these neurons, and you have kind of big chunks in there, the contrast effect dominates so much that it kind of dominates across and is stronger than the rest of the iron in the microsome 1. And that also means now measuring R2 prime in a large voxel that contains maybe hundreds of neurons gives you information about the iron in those neurons and not other things, so about 60%. So you have now a sensitive marker to microstructure that is so much smaller than your voxel size. And obviously, when you know iron concentration in neurons, that's in itself really interesting because of all the theories about iron accumulation in the neurodegenerative diseases and Parkinson. But also you can do cross-calibration because we know how high the average iron concentration in each cell is. By using and applying and exploiting that, you can actually now cross-calibrate and determine a cell concentration in the microsome. So this might be a really interesting marker for neuronal dopaminergic neuron cell loss. So we are trying to push this into the in vivo domain at the moment. But while doing that, we got a lot of flack presenting the data I've just shown to you. People said the following: look, we know we do the so-called swallowtail sign imaging in radiology, and we know it's hyper intense, it's bright. You're telling us the and we equate that with nitrosome 1 in radiology. You're telling us it's looking dark in your case, so you must be wrong. That was kind of the message we received. we were absolutely convinced we got it right because it's physics. It's basic physics, has to be right. And so we were wondering what's going on here. And you've probably seen, I mean, a lot of papers on microsome imaging, basically. It's out there, right? It's even in the textbooks. And this really bothered us, not only because we got a lot of flack for it, but it's something puzzling. And so we suspected this has to do with the anatomical localization of the microsomes. And so we went out and in a collaboration with our colleagues in Amsterdam, Anneke Alkemade, we built a really large atlas where we acquired whole brain quantitative multi-parameter map data and lots of different histological stains. And we could use that atlas to actually identify histologically microsome 1. So using the gold standard that is widely accepted. We could also use in vivo MRI, and we are really thankful to Penny Goland here, actually helped us looking at that, looking at the swallowtail sign, because they described it for the first time in the relationship to the microsome. And putting this together, these are obviously not the same subjects, right? You register them. What you can do, you can now compare this hyperintense part of the swallowtail sign with microsome 1 as defined by histology. And what you see is, actually you see the following, interesting. Microsome 1 is an elongated thin structure, and the hyperintense part of the swallowtail sign is rather like a blob. So they overlap partially, but they're definitely not the same thing. And in a sense now, simplifying it a lot, you can maybe think about it a bit like a hot dog, right? You have the microsome 1 there. There is a certain overlap, so it's not surprising that people mistook it. But we believe this is an erroneous understanding and use of the swallowtail sign. What I don't want to say, to just be very clear, we don't criticize or have concerns about the swallowtail sign being an important marker, for example, in diagnosis, but its interpretation, we believe, is simply wrong. And to help the community there a little bit, I hope this will start. Let me see. Will it? Yeah. We're also working now on microsome atlas. So Malte Bramelo is putting a lot of work into that. So we share the information of these microsome distributions, and that maybe will help with dealing with some of these conundrums. So doing basic physics and understanding contrast mechanisms actually helped us with neuroanatomy and hopefully maybe also with neuroradiology. And I'm coming to the end of my presentation. I would just like to wrap up here with a brief discussion. So I hope I could convince you that, with MRI and using the right biophysical models, you can actually glean microstructural information. And if you can build these models from first principles, and I actually forgot to mention this is really important. The model on microsome 1 and the dopaminergic neurons has no free parameters. This is fully defined. There are no free parameters in there that are fitted to any MRI data. So this is truly a physical model derived from first principles, fully calibrated. And I think if you have these kind of models, this is kind of a new domain. This is kind of a new era, how you can use MRI information. It enables you to look at structure-function relationships at a much more fine-grained level. I think there's great promise for biomarkers, but we have to get the models right for that. It's not trivial. It's a versatile and transferable technique. You can use it in patients, post-mortem, in vivo, but a lot of challenges remain, and obvious ones are better MRI quality, higher resolution, and building the right unified biophysical models that combine as many data sets in different contrasts as possible to make the estimation as reliable as possible. Validation is a big issue that gives us a lot of headache. What do you compare against? There's little data typically out there on humans you can compare against. So we work a lot on these techniques. And I think conceptually, and I think this is giving us a headache, but the entire community, we have this issue. We have the brain. When you look at it, there are 6 orders of magnitude of spatial scale involved. How do you model this? How do you integrate it? How do you bring this data together? Even if we know every cell and every axon in the brain, even if we had this information, how do you use it? How do you make sense of it? Nevertheless, I think it's exciting times. I think MRI is now moving from a, let's say, qualitative technique to a quantitative MRI technique. And we hope really to a quantitative biological measurement instrument for in vivo histology eventually. And this is a lot of work and requires a lot of different domain expertise. So we are very happy to have many super helpful collaborators, including here at MGH, that have helped so much over the years to pull that off. And obviously, I'd like to thank in particular the Department of Neurophysics in Leipzig, put so much passion and effort and time into these fantastic projects. And I'd like to thank you for your attention.